and this is about AI and drug discovery. And today I would like to discuss with you guys how it is possible to actually augment your workflows and increase their performance. For this, I prepared a little roadmap where I would like to talk about data generation in general, which can be augmented with our tools particularly. We need to take a little bit of a detour in regards to molecular similarity here, because I think this is the largest aspect that is neglected in AI. And then we would like to talk about the thinkability of compounds and how you can actually tailor it in-house or doing your searches or doing the setup of the workflow. Quite important for you guys as well, how to actually get your hands on particular compound sets of relevant chemistry that you can re-feed into your workflows. After we have talked about this whole data set generation in itself, I would like to provide you with some insights how to augment structure-based workflows. So basically post-generation, post-prediction, and how to use this 3D information to actually feed it back into your workflow to improve the quality of your predictions. By the end of the day, we'll be also talking about how to digest the data you have on hand or the data that you generated, the output. And finally, I would like to highlight two workflows that were published using our applications to give you an idea how others do that, because it's something that some people miss every now and then. Is everybody else not able to hear me though? I got like one problem, but I think that for the rest it should be fine. At least I tested it beforehand. So if anybody else is not able to hear me, raise your hand or give me a heads up. Meanwhile, the perfect outline of AI, so everybody is able to understand what we are talking about, since we also have a lot of medicinal chemists joining today. AI consists of particularly two parts to generate data, which is the output. The first thing you need is an input, and this input exists in the form of data. Data can differ into three things ultimately: the type of data you're feeding into your workflows, the quality of the data, and actually the volume, so how much data you feed into the workflow. And this is something where we can all agree upon. The more data you have, the better your results and your output will be. This is also true for the quality of the data. This is where people struggle if they just feed random data, and by random data I mean publicly available data, into the workflows. Sometimes it's not double checked, sometimes there is no form of additional filtering involved. So therefore, you always need to take at least one look into the table of your data to see, does it work out for me or not? Is everything in the right place? Is this really biological activity, or is this just CYP enzyme inhibition? So therefore, curate your data before you feed into the workflow. Subsequently, once you have your data in place, and the more data the better, right? If you have just one point, you cannot predict stuff, you cannot point into different directions. With 2 points of data, things become more clearer. And if you go into millions of points of data, obviously, the quality of your model will improve. Subsequently, you have your AI machinery, and this is where you step in, where you decide how you want to set up the whole thing. What do you want to achieve? And this is where you feed in algorithms, where you feed also the transformators, where you train the model to actually process the data. And once your model learns something, then you can also generate output. And this can be predictions in regards to molecule suggestions. Those predictions can also include the prediction of properties. And quite interesting also the 3D aspect, namely to generate valid poses of compounds at your target structure. As you mentioned before, the more data, the merrier. Therefore, in the first part of this workshop, I would like to talk about how to actually generate data. And for this, I have separated the data aspect into three regions, namely the chemical spaces, the ligand assessment, and the property and descriptors of molecules. And we will dive into them one by one. What you should always ask yourself when you're working in NI is ultimately, which data is relevant for my project? What is there to be processed? how you would like to generate this data, how to actually get more data, maybe also even stuff you are not aware of, and how to embed it into your workflows. The ultimate idea is the more data you have, the better the predictions they are, right? Yet, if you're going into especially language models, it's also quite important that you also include descriptive dimensions, right? So if you just increase the level of dimensions, you can also understand the meta level of the data, or the model will be able to extract particular details that you as a human would not uh, see by yourself. So in regards to chemical spaces, so chemical spaces are the largest source for compounds, namely for those that are tangible, that are accessible, which means that you either can purchase them or synthesize those compounds. Chemical spaces can provide you with data in regards to tangible chemistry, so molecules, but not only, you can also extract building blocks that are used in the generation of the building blocks. You can also extract the chemical reactions that lead to those molecules that you're looking at. And what's quite important, people also sometimes neglect, you can also extract the Burmese scaffolds from compounds that you retrieve from chemical spaces. And those are ultimately just undecorated core structures, pharmacophores, scaffolds that you can re-feed into your workflow to look for stuff that is really just a naked structure in itself without decorations like chlorines, functional groups and stuff like that. In regards to ligand assessment, this is the 3D world. So we're looking at structures binding at something, your protein structure, your DNA, whatsoever. And this is where you take a look into interactions between the target structure and the ligand. And those provide you with very, 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 very valuable insights of the binding mode of the ligand, and ultimately also provide an additional layer of information that you can feed back into your workflows, which is the estimated affinity of the assessed ligand, but also as well the individual contributions of each heavy atom to the overall binding affinity. Furthermore, quite important, if you, based on your target structure on the project itself, it will be also quite impactful to take a look at where are the H bonds, which are the important interaction points for quite a lot of ligands, and also the dissociation tendencies. So ultimately, how happy is my ligand and how happy is my target structure actually with the ligand? Last but not least, this is also where you can do a sanity check on poses and interactions, right? So like an reassessment of the poses that you generated to check are those valid or not. And last but not least, I think this is a rather trivial aspect. Computational chemistry is extremely strong at predicting physical-chemical properties, pharmacokinetically relevant parameters as well, sometimes. People are working on it. But nevertheless, this is where AI workflows are used, I would say, the most on pharmaceutical companies and larger companies, namely to predict the pharmacological fate of a compound once it's applied. Those parameters can include quite a lot of things, molecular weight, log P, but also the ligand efficacy, the surface area, also very fundamental things like the formal charge of the molecule, the atomic properties in itself, and also very descriptive ones like rotatable bonds and the number of hydrogen donors and acceptors. So where do chemical spaces take part of the whole AI workflow discussion? So you have your AI machinery. And in quite a lot of cases, those AI machineries are there to predict better compounds, right? They are fed with data, they provide you with predictions. Those predictions are addressed or directed to, wait, I have to rearrange myself a little bit. There you go. Those predictions are there to provide you with optimized queries. Those optimized queries can cover a lot of aspects. It can be the binding affinity or the estimated affinity of the compounds. Those can be also ADME parameters. This can also be cell penetration. So ultimately, all of those things that are part of this whole scenario of the drug discovery hemisphere can be optimized parameters. The problem is, though, are those compounds synthesizable? And this is the largest or the most important aspect when it comes down to a management perspective. If you get very nice compounds from the de novo design, so from scratch, and those compounds are good, and you have a very high success, high prediction rate for good compounds, It's cool, it's fine, but if you take 50 steps to get to your compounds, they're worthless. Nobody will allow that. It's just a waste of time. So ultimately, the question is, how can you actually um, get your hands on very nice candidates very fast? And this is where chemical spaces come in. You have your predictions, and what you can do, you can take a look into the largest compound collections, which are the combinatorial chemical spaces, and take a look which compounds are inside the space. Is it the real compound that is exactly like the one I provided for my predictions? Or also there are some very close analogs that are just, close enough to that I would say I can also test them to get an idea of if my prediction was good or not. The best thing that can happen is that you actually find something with a similarity of one, which is the identical molecule, like in this case here. Sometimes it's also possible that you find something that is just lacking a fluorine or just a metal group somewhere. I mean, like sometimes it's not that much of importance, and you can also get those compounds. Sometimes it can become also kind of like a little bit different, where a 2-fluorometal group is missing or lacking. But all in all, you do can use those chemical spaces to get very similar compounds. For this, we do have quite a lot of collaborations running. So our chemical spaces are billion to trillion size as of now. And on the right side, you can see all those companies that we are actually collaborating with to create those chemical spaces of compounds where you can retrieve tangible compounds and then just order them from those compound providers that made it into their business model to provide you with something. We are collaborating with Ambinter, Enamine, Wuxi, Lab Network, Mutara, ChemSpace, eMolecules, and also as of, it was in the summer of last year, we're also collaborating with Enamine on the trillion size chemical space, namely XReal. And what is quite important here is that it saves you quite a lot of money, right? Synthesis is expensive. You as a working person, workforce, you are expensive. Every step you are doing is expensive. Acquiring building blocks is expensive, storing them, the logistics behind them. So therefore, it's quite easier for you to just order compounds which are delivered to your table within 4 to 6 weeks. This accelerates the whole projects quite a lot, and especially those in the early stages where you are focusing on hit identification. I mean, I don't have to tell you by now, bigger is better. This is the consensus since a few years. The larger the hunting grounds are, the more and better, which is even more important, the better compounds you can find in larger compound collections. And therefore, by looking into chemical spaces, you do not get only better compounds, but a huge array of different scaffolds that you can work with. This becomes quite evident when you take a look into the nature of how AI workflows work. So basically, if you're working with AI workflows, you're providing them with actives, right? So you say, these are my active compounds, you feed them into the workflow and you request the workflow, please provide me with better compounds. So ultimately, based on your data, the workflow will generate by the end of the day, hopefully better compounds. Yet it is limited. You can only fish in a very small pond based on known actives, known structures, right? It becomes even more difficult if you don't have any actives or just a few scaffolds to work with. So what will happen, and this is what's displayed in quite a lot of publications, if you're working with de novo design, you're fishing in a very dense area around your target structures. If you would like to retrieve similar compounds, though, you need to use different methods to expand your hits, right? So what I'm telling you here is if you want to cover a larger area of the chemical space, you need to use orthogonal search methods that exceed this trivial area around de novo designed compounds. And those compounds, as you will see in the upcoming slides, are also very similar and actually quite efficient to expand and refeed into your workflows. What I did here on the left is to actually highlight the um, distribution of different search methods. So basically, all of them are command line applications, search methods that we have developed in-house, F-Tree, Spacelight, and SpaceMax. And by starting with the original molecule, we used all of them. And then, after having retrieved 100 results for the original molecule, we actually mapped them with a UMAP. So basically, for the medicinal chemist, what is a UMAP? A UMAP is some sort of dimension reduction technique where you take a lot of dimensions and reduce them into 2 or 3 dimensions. In this case, it's 2, to get a feeling for how clustered are my compounds, right? So this uses a molecular fingerprint method, easy as before, and now it compares the different results. And you can actually see that each search method provides you with different clusters of compounds. So ultimately, each search method provides you with new scaffolds that are still similar to initial compound. And what's very important, this is where what you have to engrave into your heart and mind, um, those structures, especially F-trees and spacemacs, and also the new fingerprint method that was developed for spacelight, FCSF4, they all retrieve stuff that you would miss otherwise with other methods. Okay? So basically, what you would expect with de novo design is a clustering around the compound in this zone. Fingerprint methods, ECSF4 is able to extend it to some degree, but additional methods cover more area of the chemical space that can lead to new structures, new scaffolds to be confirmed by additional computations to be actually of value. So therefore, to leave the same pond for de novo design, apply new methods. This is something I have taken from a publication that did a very similar thing. So basically, in green, we see the cluster of de novo designed compounds. And then what they did subsequently, they expanded their hits based on F-trees. But you can actually see quite nicely how you're able to expand into different areas of the chemical space by doing so. Why I'm telling you this ultimately, and this is what I have mentioned in another webinar quite like 1 year ago. So de novo design AI lacks novelty. So all of the things that emerge from AI do have some form of limitations. Things improved, I have to admit, things improved quite a lot. Yet still, especially in the beginnings, if you don't have, your workflows established, you're not that experienced, and so on and so forth, you lack novelty. So all the compounds that de novo design creates is something you can up by yourself or by just applying simple medicinal chemistry transformations. And this was also shown for quite a lot of things, where also the FDA approval of AI-generated drugs was not provided or not given because it lacked novelty. As mentioned, to get chemical diverse compounds, you need different ligand-based methods to search for them, which I will show to you in the next slide. And what's quite important, if you use orthogonal methods, you can actually let them double-check each other. So use method A to confirm results of method B and method C to confirm results of method A, and so on and so forth. So basically use those things to the results of each other, all search methods, to enrich similar compounds, right? A compound that is considered similar by all 3 different methods, or at least within a certain, margin, is quite likely to be very similar to your initial structure, to your to your starting point, which also means it is very highly likely to be biologically active as well, right? So ultimately, to enrich better candidates, just use all methods at the same time. So what are the methods that I've mentioned? So basically, I don't want to lose too many words here. We have F-trees for fuzzy pharmacophore matching. We have Spacelight that performs fingerprint-based similarity searches. ECSF4 is the default, but you can use also other fingerprints. And ultimately, the maximum common substructure matching tool, SpaceMax, which takes into consideration the longest chain of ongoing heavy atoms compared to your QE structure. What can you expect? So given our initial compound here, which is an adenosine receptor antagonist, as far as I can recall, these will provide you with compounds that have the same pharmacophore flavor to them. They are like very novel IP, quite nice for scaffold toppings, basically trying to match pharmacophore features like H1 donors, aromaticity, and so on and so forth into the structures, which can lead to very diverse scaffolds, which I personally really like. Not always that valuable if you're working with a very particular target that really needs exactly this pharmacophore. Some people don't like this pharmacophore fuzziness to the tool. I personally love it, and I can show you in the upcoming slides what I mean by that. Close analogs not so surprising, can be achieved with fingerprint-based searching methods, namely spacelight. Are quite efficient if you perform structural activity shape analysis, so close analogs with just smaller variations of decorations. And ultimately, MCF search quite nice if you would like to have the exact framework of the compound in itself, basically meaning you will end up with very similarly sized compounds if you perform a search. Also, not so relevant in this case for AI though, but can be still mentioned. You can also perform exact substructure searches, which are extremely important for fragment-based drug design. Okay, so now diving back into the UMAP figure from before, I used the screen molecule and looked into one of several chemical spaces and retrieved compounds. So now we have 4 different rows, which each for each search method, one for F3 spacelight EC34 and spacelight FC34, as well as spacemax, to just give you an idea about the similarity of the results and also the chemistry that comes with it. Here I have each picked the top 3 ranking scaffolds, or the top 3 ranking results from a search of 100 compounds. And then what I really like about the F3 results, they really try to match the theme or the flavor of our query compound. We do get the alignment of several ring systems, 1, 2, 3, 4, 5, 1, 2, 3, 4, 5. So ultimately the same ring numbers, the same ring number. We get also the hydrogen bond acceptors in this area and also the fluorine back here. So ultimately, this is something which you can see also in the calculated values for the other search methods. So I double checked everything with the other methods as well. And you can see that the ECSFP score drops quite significantly. And if you ever performed an ECSFP search, you will notice how sensitive it is towards particular decorations like halogens. So therefore, this is a compound you would never, ever see with ECSFP4 given the size of the chemical space. Yet, if you compare it to the results underneath, you will see, well, from a flavor, from the pharmacophore flavor, from the perspective of 2G similarity, I would say that this is something that can actually bind to a target. So we do see quite a lot of similarities here. In the case of the ECSFP searches and fingerprint-based methods, we can actually see that we are lacking quite a lot of molecule at the terminal area, which can affect, obviously, the biological activity of the compound. So what I'm telling you in this case is that here, F3s was the best one, yet it was the one most further away from the initial compound from a cluster perspective, given the ECSFP4 fingerprint search method that was applied for the dimension reduction of the UMAP in here. Another example, a small molecule, also biologically active. In this case, you can actually see quite nicely how the fingerprint-based methods and the maximum common substructure-based methods really do cluster around our compound of interest. We do not find the exact same molecule, the exact same scaffold, which is okay, which is fine. In this case, F3 though is very far apart from what we could expect. So again, F3 is also what I always tell people. Those are apples and oranges, right? So we have different fruits here. You cannot compare those numbers. 0.961 in F3 score does mean something completely different than 0.9 something and fingerprint methods, right? So the F3s usually ranges from 0.8 to 1 point something. So the similarity here is always quite high. Fingerprint-based methods, the range is something like 0.75 is still considered very similar to initial structure, and 0.3 is usually the cutoff that I see companies being used, again, depending on the case of the project. So therefore, the numbers are very different for each algorithm and should be also handled like that. Spacemax, depending on the case again, 0.6 is something I would still consider quite high. Now again, to the molecular features. Here in those cases, we can actually see that we try to match the hydrogen bond donor character in this ring system. We do have a flip of the amide group though, if this is essential for the interactions of the amide. this will be a failure. So I would say here the F-tree similarity doesn't count, as F-trees doesn't provide me with very nice compounds though. Yet for the other cases, we do come up with very reasonable suggestions for similar compounds, which includes the fingerprint-based methods and SpaceMax. So this is an example of what that I did with 12 compounds, just to provide you with an idea of how well each method actually complements each other to cover the chemical space. But you can see quite nicely that every Cree molecule gets its own cluster. So very separated from each other. And each method is actually able to provide something in the periphery of our Cree structure. So now if you just think about, okay, let's remove F-trees, the dark blue ones, or Spacelight, FCs before, the orange ones, you would see how those clusters become less dense and kind of like just focus on a particular spot. So to get diverse compounds, I recommend to use more than just one method by the end of the day. Tool handling from our side. So since this is, I would say this is a comp camp heavy workshop. So therefore, I don't want to dive into the GUIs of Caesar or Infinity. Yet all those command line tools, Spacelight, F-trees, SpaceMax, are very easy to handle from the command line thing. It's very straightforward. If you put in the help functionality, you get a list of what is requested and what you can actually modify. If you still struggle to understand every single aspect, we also have very sophisticated user guides that provide you with additional insights how to operate the command line tools efficiently. Okay, so now a little bit more focus on how to actually implement those tools for AI. This is, I would say, the most neglected aspect that people don't address. It's a tautology, but it still works. Ultimately, chemical spaces are there to be your providers for customized libraries. You use seeds. The seeds can be active compounds. It can be just made up stuff. Doesn't matter. And use them to actually go into chemical spaces and mine for custom libraries. So basically, you apply the search algorithms, and you can request up to 1 million search results per query compound, which helps you to really construct very tailored custom libraries to refeed into your workflows or to use in other applications. This works quite nicely, and this works also quite fast. I mean, like enumerating 1 million molecules takes its time, to be honest, but you can also work with smaller batches, 100 compounds, 10,000 compounds, 50,000 compounds, completely up to you, to create chunks that are handleable by your workflow. What I also quite like to mention is this is not where it ends, because if you generate those compounds come with additional info, which will be shown on the next slide, that you can use to orchestrate your workflow even further. And last but not least, if you actually mine for tangible compounds, you can use also the compounds that you generated to generate Grimm's scaffolds, which are just the undecorated versions of your compounds that you can again refeed to see how you can improve them further. Okay, now to take a look how you could actually do it in-house to fine-tune your own chemical space. You only need two things to create a chemical space: building blocks and reactions. Mentioned in a lot of webinars by us. If you have more questions, reach out to me. We'll go into detail here. Once you have your chemical space, this is where the fun part starts, because you can use the chemical space and improve it, make it evolve. So if you start tracking the reactions, how good was reaction A1? How good was reaction AB2? And so on and so forth. You can actually then start to modify and adjust those reactions. By modifying the smarts and smirks, you can actually say, okay, I don't want to include those kind of building blocks because they just don't work for my reaction. This is what the compound providers do. So Enamine, ChemSpace, they all apply machine learning algorithms to fine-tune the predictions of the synthesis to increase the synthesis rate and to actually streamline things, not to waste weeks on a single compound, but to create a compound within just a few hours or a few days. What's so important about this one is that you can define reactions that are extremely efficient, but they can be done within half an hour, including the purification. Also, what's quite possible is that you can actually flag reactions that are important and provide you with valuable compounds, but require additional effort, which can slow down the process. So what you can do then is to actually map stuff. And this is something that management cares about quite a lot. How fast can you synthesize the compounds? Which compounds or which reactions should be avoided? Which ones are expensive even? And are there better alternatives? So ultimately, if you have your chemical space running, you can also identify particular aspects that I've mentioned, costs, time, availability, and provide them as tags to your building blocks. So the output of each chemical space is a compound ID, including the reaction and the building blocks here. And by modifying the name of the reaction block, like C1 is inexpensive, C2 is moderately expensive, C3 is extremely expensive, you can just take a look at the ID tag of the name of the molecule that came from the chemical space and see instantly, oh, okay, C3, this will be a very expensive compound. But you can do as well encode some form of distance to purchasability, right? So C for cost, but you can also use D for distance. You can say, okay, D1 means this is an in-house building block. It's already in stock. It's on the shelf. I can use it just right ahead. I don't need to order stuff. D2 can mean that it requires building blocks from the vendor that you can also use as an input data for the chemical space, which means it would take quite some days, maybe a few weeks, until the building block is delivered to your table, and then you can start the synthesis. Furthermore, you can also use it on the reactions where you say, okay, this is a very complicated reaction. The purification is a nightmare because there'll be a lot of stereocenters. I don't know. So therefore, you can also encode additional effort as a tag into your reaction to see, okay, this is a reaction that we should avoid and only use every now and then if the compound is really promising. So if you also have those kind of things, you can extract those information, basically compounds, reactions, and building blocks for promising candidates that you can refeed into your workflow to increase its performance. Also, obviously, since you can work with building blocks, I mean, also building blocks can be an argument where you say, okay, I just want to know, I don't know, use 15 building blocks for whatever reason, or because I'm using like automated synthesis in-house. I just want to focus on like 90 building blocks. What compounds that I retrieved are containing those building blocks, or what building blocks should I have always on stock to generate those compounds? So ultimately, it's not only the compounds, but the whole environment. It's basically the whole infrastructure around compound synthesis can be encoded inside the chemical space if you're doing it in-house. Okay, let's summarize what we have learned so far. The bigger, the better. More compounds, more possibilities, which means more data points to feed into your workflows. To be efficient and cover more chemical space by searches, you need orthogonal screening, which means please use our screening algorithms to retrieve more compounds that cover more of the chemical space that is available, that is also tangible. The chemical spaces can be used for the custom library generation. And this is something you can also guide, right? By using only actives, you can also just generate compounds based on known binders, which also accelerates the whole process quite a lot. Or you use diverse compounds. What we do in-house, we do have a set of a very diversified set with a high focus on biological activity and biological relevance that we actually use as seeds to retrieve relevant chemistry from chemical spaces. Cost control, management loves cost control. It's something that kind of like gets lost if there are a lot of things involved, a lot of libraries, a lot of systems. A chemical space can unify all of them inside one thing, inside tags that you then can use to really assess everything. Tangibility of a compound, how long it will take to get your hands on it, and so on and so forth. And obviously, more data points for decision making, you get more factors. Similarity of compounds that are not only fingerprint based, you get the compounds, the SMILES code, you get the distance to purchasability, as well as additional molecular descriptors like, all those physicochemical stuff. So far, so good. Now to the second point. The next 2 points will be a little bit shorter, I hope at least. Now into the structure-based area. So ultimately, Again, I would like to highlight a command line tool here, namely hide. And hide is our scoring algorithm, not the docking algorithm, the scoring algorithm. So what it does, it takes a ligand, it takes a target structure, and estimates the overall binding affinity of the ligand. So far, so good. What people are not always aware of, that each individual heavy atom's contribution to the overall binding affinity is actually calculated. So ultimately, we know exactly, at least according to our algorithm, how much this nitrogen contributes in this context to the overall binding affinity of the ligand. So the trivial application is here to use hide to just double confirm your post generations. I mean, most workflows in the structure-based context use some sort of docking predictions or docking step to provide a docking score that is then used as a metric for the enrichment and so on and so forth. It is always a good thing to have a second partner, right? One algorithm is blind. It may be wrong, it may be right. But if two scoring algorithms agree that this is a good candidate, obviously it will enrich the hit rates. This is something that you can obviously use just a metric to feedback into the workflow in itself. But, and this is where the fun part starts, we also want to learn something today. There's also an advanced use case. So while operating hide, you can use a very interesting command, namely the write atom scores command, which will provide for each ligand an additional set of descriptors, namely the individual contributions of each heavy atom. And this is in a CSV format. So this is exactly from this particular ligand. And I use this command to also write out the atom scores. And what I receive is a matrix giving me ideas about the total score of the heavy atom in regards to the receptor. How satisfied is the receptor with the atom being there? Zero means it doesn't care at all. It doesn't gain any binding affinity. So it didn't dilute it with water as well. And also those considering the ligand, namely the dissolution, free energy that we gain from it, and the interactions. And we do it for each of one of them. So this can help you to identify hotspots, right? So basically, this can help you to say, in this region, nitrogen is quite important. In this region, it's the carbon atom. I'll also show you an workflow subsequently. And what I really like about this one, by using hide, you get a lot of additional informations that holistically cover your complex, which means you get also information about the quality of the torsions, the inter and intermolecular clashes, the number of hydrogen bonds, and also the estimated affinity of your ligand. So how do people use it in motion? This is one example that is dedicated for preparation of structures. What hide does, hide also provides, or not provides, it provides coordinates for hydrogens for each PDB structure or each 3D structure that you feed into the command line tool, which can be used to refine the hydrogen bond network around a compound of interest, calculate hydrogen bonds, and also it's also possible to really fine-tune the best pose of your ligands. So minor adjustments are allowed if you use hide. You can also turn it off by using this command here, hbound only, which can improve the initial pose of your complex. And if you're doing virtual screenings, especially template-based, or if you grow your fragment, it's of utmost importance to have the best starting point to work with. And hide is extremely well in actually generating those kind of things. So you're starting with a computing complex, you can apply hide to calculate individual contributions, but you can also use hide to optimize the H-board network involved. If you don't want any additional adjustments of poses, like it should remain exactly as it is, every heavy atom should stay in this 3D position. It's also possible with this command, so therefore no hide scoring will be applied, which means you will not receive the individual contributions and no pose adjustments, but you will get at least the hydrogen bond network that may be of interest to you. How do others do that, or how could others do that? Um, or how do others apply that? I really liked this workflow that I've seen. Basically, starting with molecular dynamic simulations, this company tracked the trajectories of the conformational changes and then used them as input. So basically, they extracted snapshots from the MD simulations of low energy conformations and then ran height over it. Why did they do that? They got additional insights of layers of information, estimated binding affinity, clashes, and so on and so forth, which can help you to understand the full picture, right? If you're just working with docking predictions, it's a snapshot, right? It's exactly this conformation of your ligand. It's exactly this conformation of your um, target structure. You don't see the behavior. You don't see how hydrogen bonds may switch, may swap. Are those even hydrogen bond interactions that are actually present all the time there? So by doing so, you can also use height to assess those complexes to actually check for recurring patterns or to identify unique events that actually just not really contributing to the overall binding affinity in the larger picture. Obviously, those are all things that augment your decisions when you are performing subsequent drug design. What's next? It's the model training. So what you can also do for height with height is to start with your complex, apply height, and then extract those details about individual height contributions. Where you say, okay, I get those contributions for this nitrogen. So it seems like the hydrogen is not very happy there. No surprise, because we have lipophilic residues here. You can do it for both the ligand and the receptor, ligand and receptor, to actually mine for those kind of informations to see, okay, in this position, I would like to have a nitrogen, fluorine, whatsoever, to then do the de novo design with those kind of informations to assemble your molecules. Just to mention a few other use cases that I have encountered over the years. Obviously, people just use heights just really to supplement their structure with hydrogens. It sounds boring, but people do that, especially in the I workflows, because they need fast preparation of structures and also fast assessing, reassessing of hydrogens and protonation states within the ligands, within the complexes, which can be done quite nicely with height. Orthogonal screening methods for hit enrichment, doing a lot of dockings, having like a second opinion with height is also quite nice. The refinement of poses for template-based docking and fabric extension. The sanity check that I have mentioned. So basically, if your workflow is there to really do de novo pose predictions, it's quite nice to have somebody like Hyde looking over it and tell you, well, there are no clashes because your algorithm didn't include them, just to like really see, is this a valid pose or not? Or are there a lot of crashes? Are there a lot of torsions even? What I've seen and I found very interesting is the surface sampling of target structures for poorly assembled regions, which can also help you to use like a ligand and place it at the target structure to see, okay, there is a lot of lipophilic area that is not satisfied or can be better satisfied by lipophilic ligands, which means that this target structure is maybe not in its right conformation. And we should do another MD to really refine the structure or the model we are working with. What I'm seeing quite a lot that those AlphaFold associated algorithm AI workflows are now starting to also supplement with water molecules, cofactors, so all that stuff that is usually co-crystallized in PDBs, and become quite nice with that. And obviously, height can also take them into account. So this is something that works quite nicely if you are using those workflows, those models. It's quite nice that height also takes those kind of things into account if you wanted to. A very short slide. Properties and descriptors. I mean, you can calculate stuff. This is a list of stuff that is associated to the height algorithms. You can also even use FlexX to generate more docking scores. So anything structure-based is in green, and all of those ligand-based 1D parameters whatsoever are mentioned in dark bluish. So basically, you can use our tools to calculate those kind of things for dimension reductions, right? Why do I mention that? All of those fancy things, UMAP, PCA, principal component analysis, TSNE, are used to cluster compounds, which can mean a lot of things. You can cluster compounds based on activity. You can cluster compounds based on toxicity, based on atomic property, cell penetration, you name it. And the more of those compounds you have that you can associate with members of each cluster, the more likely you may end up with clusters that are actually nicely predicting the outcomes of results. So ultimately, if you're performing dimension reduction just to really, navigate within your generated output data, it's also always valuable to have additional layers of information that you can feed into the dimension reduction. Okay, the last few slides are dedicated to two companies, or a half companies, three companies, that published actually workflows using our tools. One is Millipore, Sigma, and Merck, and the other one is Orion Pharma. And those are publications. So basically, I'm allowed to talk about it because it's now public knowledge. And I would like to highlight how they did stuff. So ultimately, what Millipore, Sigma, and Merck did in a collaboration, they created the Edison platform for the generation of compounds that can be synthesized, purchased as a service. So you can just go to Millipore, Sigma, select your PDB, and let the whole workflow generate compounds. And then you say, okay, I would like to purchase compound 5 to 10, another 100 here, another 600 there. And what Millipore, Sigma, and Merck did here, they used actually F-trees for hit expansion. And they were actually able to show that F-trees was able to extend the de novo designed compounds and increase the diversity here. I'll talk about Orion on the upcoming slides. So basically what Millipore Sigma did, this is the workflow that I have modified a little bit to make it more clearer. So basically you start with input compounds and then you just use reinvent for the de novo design of compounds. I think most of you guys know reinvent. I don't have to waste my breath here. What happens then is the 3D model evaluation by docking. So basically the compounds generated by the de novo design are then docked into the binding site. And once you see compounds that are actually valid or promising candidates, this is where the fun part starts, where we also join them, namely by augmenting the hit extension with the chemical spaces. So all of those compounds that are actually confirmed as quite relevant, interesting, promising, whatsoever, are then taken into the chemical spaces, and then results of similar compounds are then retrieved with F-trees to expand the chemical diversity here. Once it's done, we are at the retrocensus part, run by Cynthia, which actually just double checks for potential costs of the compounds while it dives into the ultra large chemical spaces for reactions and everything. So this is what they did. This is a use case that they have also mentioned in their publication. They ran a case study on tanquinase inhibitors. The challenge here was that a lot of scaffolds were available for the target structure, and they just wanted to really show how they can come up with new scaffolds, right? And I think this is quite important, especially for the pharmaceutical industry. So the workflow is, as I've mentioned, they started with the de novo design, then docking, and then based on the promising hits, they did the hit expansion with the real space, Galaxy, and their own chemical space that they have generated with our software, the Sigma Aldrich space, the SA space. And they also augmented the whole thing with some other additional 2D search methods. And what they were able to show, they were actually able to not only find known binders here, that are chemically similar. Yet, if you would look at that from a fingerprint-based method, I would say not even close. And they were actually able to expand the whole array of chemical diversity by applying the method. The second example done by Orion Pharma, also a very interesting workflow designed for screening ultra-large chemical spaces. Just to be faster, ultimately. On the left, the original workflow. On the right, my stylized interpretation and modification. So basically what they do, they start by docking seed compounds in an enumerated library, and then apply their docking scoring algorithm here. Once they found good candidates, they again use those seed compounds here, and then search the chemical spaces for close analogs, similar compounds that are again reassessed for their docking score prediction. Then the best compounds are then really redocked once again. So this is the hard docking. This is the machine learning augmented rescoring. And by the end of the day, they end up with a virtual hits for the triage. And what they were actually able to show in their results is that the two methods they use, spacelight and F-trees, are both providing them with diverse compounds, diverse scaffolds. And the overlap between both methods is not as huge as expected. So only a small margin here remains that can be found in both search methods. Yet it makes more sense to actually apply both methods as well. What I also really like, and this is not highlighted enough from my point of view in the publication, given the tree structures that they investigated, they were able to retrieve a huge amount of novel scaffolds compared to the ones found in Campbell, and only a smallest fraction of them were actually also considered were the same ones. So basically, really for finding new hit compounds in a computational environment, quite efficient workflow, and I have to admit that from a CompCam perspective, it's really nicely done. Just to highlight some interesting facts here, they also compared their workflows to random docking, and they did the workflow for their space has workflow for 3 million compounds, and then brute force docked 3 million random compounds from the enamines real space. And what came up with by doing random docking, only 6 virtual hits would emerge, whereas in their case with space hasten, they were able to retrieve almost 40,000 virtual heats based on their algorithm. So huge discrepancy here by doing the brute force method compared to a smart approach.